Okay, so now let's talk about when things go wrong with insulin secretion or the pancreas. And the first major problem that we'll talk about is type 1 diabetes. So I'd like you to be able to define what diabetes is and compare and contrast the differences between type 1 and type 2 diabetes. And I will also say that is also one of my favorite exam questions. I want you to be able to describe and explain the etiology and pathophysiology of type 1 diabetes, explain the symptoms and diagnosis, explain current treatments which are available, and then talk about the challenges of encountered for each treatment, what the problems and drawbacks are. So first we'll talk a little bit about history and definition of diabetes. We'll cover epidemiology, and then we'll talk about diagnosis and treatment. So diabetes, the term, the word first was used around 250 BC. So over 2,500 years ago. The term diabetes is a Greek word, and it means to siphon, like a straw. So when you've got a nice bubble tea with the straw, right, you're siphoning the bubble tea up. So the same concept applies here, where it comes from the idea that diabetes seems to rapidly drain fluid from the affected individual because they are constantly urinating, and this leads to symptoms. So they're moving, a siphon is moving like a straw, one liquid from one to another. And this is to siphon. Symptoms include, due to the siphoning, polyuria, which is excessive urination, secondary polydipsia, which is excessive thirst, you're always thirsty. The complete term, diabetes mellitus, was coined in 1674 by Thomas Willis, with mellitus meaning honey, and that has to do with the fact that the urine has excess glucose and high levels of plasma glucose, which exceed the renal threshold. And then about a little over 100, in 1889, a little over a couple hundred years ago now, is the German physician established a link between the pancreas and diabetes, von Mering and Minkowski. And then as several of you did in your posters, and we heard in these wonderful presentations this week, and I'm sure we'll hear more about, is the first patient was successfully treated with insulin, and the discovery of insulin itself, the hormone, was a major, major breakthrough and a major contribution to science. And then many derivatives and breakthroughs have continued to be made across the globe, including, as we heard, in China. And again, this is Banting and McLeod awarded the Nobel Prize in 1923. So diabetes is important to remember. It's a group of diseases, okay? It's a little bit like cancer. It's not one disease. It's actually a group of diseases, and they're all characterized by high levels of blood glucose resulting from defects in insulin production, insulin action, or both. So it's defined as a metabolic disorder of multiple etiology characterized by chronic hyperglycemia with disturbances of carbohydrate, fat, and protein metabolism resulting from defects in insulin secretion, action, or both. So what is the epidemiology and the symptoms? Just in the UK, this is from a few years ago. These were the numbers, and you can see it's in terms of type 1 diabetes, it's only about 10%. And this percentage generally holds, it's between 8 and 15% globally across the world. In general, for diabetes, the estimated prevalence globally is around 500 million people now. And it's expected to be 10% of the population and by 2040. But there are populations that are incredibly high in diabetes cases in the Middle East. There's certain countries that have very, very high diabetes. And my father is a physician in Texas in the United States, and where his region is, they have diabetes. Over 50% of the population is diabetic. It's a tremendous, tremendous problem. And then it's thought that 1 in 2 adults with type 2 diabetes are actually undiagnosed globally. So it may be actually a bigger problem than we think. And as you can see here, there are numerous countries. This is now an older slide, but it's probably more now, with over 10 million people with diabetes. And I think someone put up on their slide what the prevalence was of diabetes in China. Now I don't remember what the number is, but I'm sure it's a significant number of people. And that's why research is very, very important. In fact, I remember when I was in Shanghai at the science park outside in Suzhou, and one of the major companies in Suzhou was looking into insulin and diabetes type biotech. I can't remember the name of the company right now. But importantly, it affects people in both urban and rural settings worldwide, with 65% of the cases in the cities. A lot of this obviously is going to be due to lifestyle, and we'll talk about that. So this is an important slide for study purposes, comparing and contrasting type 2 and type 1 diabetes. And type 1 diabetes, importantly, its onset is primarily in childhood and adolescence, and we'll talk about why that is. Often the people are thin or normal weight at the maximum. They're prone to ketoacidosis. They are insulin administration is typically required for survival. We'll talk about why that is, and that's because the pancreas is typically gone. The key beta cells are damaged by an autoimmune attack or something else, and so they don't have any way to produce insulin. And importantly, it's quite important that this is diagnosed quickly in young people, because it starts to be life-threatening. And the typical treatment is insulin injections, and obviously there's a genetic component to this, as well as an HLA antibody component. For type 2 diabetes, we'll talk about more about that in the next series of lectures, but the onset is predominantly after 40 years of age. Typically, the people are obese. They don't have ketoacidosis. Insulin administration sometimes is required, but not always. It's certainly not usually for survival. Pancreas is typically not damaged by autoimmune attack, and there's a relative insulin deficiency and or insulin resistance. Treatment is either a better diet or increased exercise, hypoglycemic tablets, or insulin injections. And you get increased prevalence, obviously, in relatives, and identical twins usually above 70% concordance with no antibody association. So type 1 diabetes, as I mentioned, also known as insulin-dependent diabetes, is typically early onset with 5 to 10% of diabetics being type 1. These guys are dependent on insulin injections from a very early age because of the requirements of insulin to the body, all the things we've talked about. And then WHO, World Health Organization, predicts 30 million patients by 2025 with, it's commonly caused by autoimmune destruction of beta cells in the pancreas. And this is just showing you, again, a picture of beta islet cells, and then I just want to use that to highlight, we talked about the insulin creating the beta cells and the glucagon in the other cells, but this is what happens when you get an autoimmune attack into the pancreas and destroying the cells. And you can see all these tiny immune cells come in and infiltrating and attacking the beta cells and killing them. And this is why you get type 1 diabetes. And here you can see gradually this is staining now for insulin. This is a pediatric control, and you can start to see as in the type 1 diabetic control, you start to get a lot less and less insulin production. Yet glucagon, the glucagon cell producing cells are maintained. So in terms of genetic predisposition, you have a mix of mother and father influence, with obviously twins being a major problem if they're monozygotic, and all of them associated with HLA antigens typically. It's thought that basically what happens are islet cells get damaged maybe by an infection of some kind. This triggers release of insulin pieces and or other cell protein fragments that get recognized into the lymph node. Then this leads to an immune response to these and an autoimmune reaction of T cells which attack the islet cells. And eventually selective beta cell destruction and T cell autoimmunity. And then there's some debate, obviously, if there's an environmental trigger. It's obviously some of this can be viral, which we'll talk about. You can get this, what happens is the lymphocytes infiltrate, and often this is just staining with anti-CD45, a marker for lymphocytes, and you can see you have these immune response just demolishing and destroying the beta cells, causing an apoptosis. So what are some potential triggers that are known? One of them is Coxsackie B virus. I'm sure there are other viruses that can also trigger just a general, major viral infection at a young age, but Coxsackie is one of the known ones. It's thought that cow's milk can also trigger it. Bit unclear if this is raw cow's milk or pasteurized, but can be part of the issue, and it's unclear what it is in cow's milk. It's obviously not everybody. Plenty of us drink cow's milk. It's clearly something specific within there, maybe a bacteria related, but again, unclear. And then in some rare cases, there's been connections to childhood vaccinations, but this is very, very uncommon. And mumps, which is also a viral infection, can also be an environmental trigger for type 1 diabetes. So in the case of Coxsackie B viruses, you get infection as a child, then you're going to get persistent infection of the beta cells of this virus, and then expression of interferon alpha in the beta cells, which will then lead to an MHC class 1 response, and recruitment of macrophages and T cells, and eventually destruction of beta cells by the activated macrophages and T cells. And then part of this destruction could then lead to an additional immune response and memory response against pieces of your beta cell cells. So it could be pieces of the receptor or some other specific marker that would then lead to chronic destruction of beta cells. Another one is can be from enteroviral infections. So certain enteroviruses are specific for infection of beta cells, and you can see here this is staining in red of the enterovirus, and you can see that for some reason these enterovirus can enter into these beta cells exceedingly well, and you get co-localization with insulin and your enterovirus. But they don't seem to impact the glucagon cells here. You can see here, this is glucagon down below in green, and it's not affected. And for some reason, something about those beta cells is facilitating infection, and you get destruction of your beta cells. Again, just showing co-staining of using insulin and co-staining of insulin in enterovirus infected pancreas, and you have very, very low production due to destruction of the beta cells. So just possible sequence of events in islets and type 1 diabetes, and there's obviously a large interest in trying to understand all of these, but this is sort of what we know so far. But all of it leads to the bottom line from our standpoint is you have some sort of destruction of beta cells and dramatic drop in insulin production and gradually autoimmunity against beta cells and eventual insulitis and insulin deficient islets. The timing of this varies somewhat, but it tends to be obviously under the age of 5 or 10. And you can see that it takes time. Because it's an immune response, it's sort of gradual. You get some sort of precipitating event, and then gradually over time you get normal insulin release and less insulin produced. You sort of have this pre-diabetes period where you start to see C-peptide present, progressive loss of insulin release, and eventually you completely lose production of C-peptide. Remember, that's the little peptide that's produced along with proinsulin, and eventually onset of diabetes. Symptoms of type 1 diabetes are lethargy, polyuria, polydipsia, candida infections, blurred vision, weight loss, ketosis, ketoacidosis, and if not treated, obviously, these days it's less common, but certainly in the old days, the child would often die. These days we know what we need to do, and so that's why in many ways the Nobel Prize was so important, because it saved many people's lives. All right, so that's kind of covered the mechanism and sort of the history and etiology of it. Let's talk, and symptoms, let's talk a bit about what we do to treat these people. First, obviously, you have to diagnose them, so you know to know the age of onset, its rapidity of onset, the phenotype, recognize the weight loss, the ketosis, recognize the insulin antibodies, all the different things that are antibodies against other parts of the beta cells that are common, one of them being the zinc transporter, and then be able to measure the C-peptide, the drop in C-peptide over time. So one of the things that we can think about and measure is are the ketone bodies. Okay, and just a reminder, ketones are a part of the metabolic pathway. So you get acetone production or D-beta hydroxybutyrate production from acetoacetate, and you can measure this ketone levels as a key readout of the metabolic state for the patient. Another one that we've talked about is the C-peptide, and this is probably the more common way to, or easiest way to measure it, is look for whether or not C-peptide production is dropped. Just a reminder that the C-peptide is co-translated, processed, and secreted with insulin. So it's an easy and nice way to measure drops and changes in insulin production. But prior to the C-peptide test was ketone bodies were followed. What about treatment for these patients? So the major treatment is still today in some form or another is insulin therapy, either through manual injection or typically these days often through pumps, which is sort of an automated. There is some interest still in beta eyelid transplants and to a lot of interest in sort of stem cell, some sort of cell therapy for helping patients where possible. And then the question, obvious question is why injections? So why can't we take insulin delivered orally? And there's a lot of interest in trying to do that. And why is that? So that's a drug discovery question you can think about. But remember, it's a peptide degradation, unique glycosylation. Will it make cross? Can it pass through the gut, etc.? Remember, we learned from our wonderful colleagues this week in our poster that in 1922 you had the first, with 21 and 22, the discovery of insulin, and then the first treatment with children. Commercial production then started almost immediately, with the primary sequence of the amino acid actually not coming until almost 30 years later. This identification of the amino acid sequence then facilitated artificial production of the hormone using E. coli, and then the commercial production started not until 82. So almost 60 years after the discovery of insulin, we could make an artificial synthetic analog. And what the advantages are, well, I'll explain to you what the advantages are of these synthetic, is you can get short, intermediate, and long-acting insulin preparations by just making different versions of the recombinant form, with slight modifications that can impact solubility. It can impact turnover and protect against degradation, and obviously also on and off rate of the receptor. Soluble insulin is used typically for emergencies in diabetic ketoacidosis, whereas an intermediate preparation is often combined with soluble insulin taken before meals, e.g. isophane insulin. And then long-acting insulin is a different preparation which can be used in different forms of it for longer periods. As we heard from our colleagues on this wonderful posters, that insulin was first identified in cows and pigs and purified. And importantly, there are some amino acid changes that were not appreciated until we knew the amino acid sequence. And you can convert pig insulin into human insulin by just removing one amino acid that distinguishes them and replacing it with the human version. Now, just a reminder for the recombinant technology, I'm trying to remember, none of you did in your projects any protein. I don't think you did protein purification. I guess some of you did. Some of you did the yeast projects, but no, I don't remember anyone actually purifying protein. But essentially what they do for this recombinant technology is they take a recombinant plasmid, put it into a bacterial cell, use the bacterial cell. You can, in these days, they use also mammalian cells and all types, but originally in the first production, in the case of Humulin, they used E. coli, make the protein, and have it serve as a factory for the enzyme. And then later this was done in yeast, and then later obviously done in human cells. Now, by using these different modifications, the Humalog, the Novolog, you get faster action. This Lantus version, you get slower action. Again, just different recombinant versions of insulin that can be tweaked for the different effects that you're looking for. So this is just a good slide reminding you the combination of all the things you've learned about and how the simplicity of lab work can really lead to a nice human drug that could save a lot of lives, and nice translational example. So you can take DNA from the human pancreas cell, put that in a bacterial DNA or yeast, make this recombinant bacterium, ferment it, grow it, purify the protein that's secreted in this human insulin, and then use that in a preparation for as human insulin. So what are some of the different types of insulin? Again, depending on what you're after, if you want fast acting, you want slow acting, you want long acting. Typically, human insulin is what's considered regular insulin. It's on the market in the U.S. called Humulin. It's made from E. coli or from yeast. It's the pre-mixed insulin, so that one you still have to mix. It's served as a powder. The pre-mixed insulins come with this zinc added, which just provide a little more solubility. Insulin analogs, which are probably more common these days and fancy, have fatty acid acylated peptides. And Humalog came out with an insulin Lispro, insulin aspart, insulin glargine. These are all modifications that are done on the peptide. And then eventually the hope is to develop some sort of orally available insulin. I know Novo Nordisk is working very hard on this. As I mentioned to you, a key aspect of these synthetic insulins and of interest is the different duration of actions. And alterations in the amino acid sequence are also used to speed absorption. So by certain modifications that are done, highlighted here in these bubbles, you can alter by here. This ASP, for example, they just added an aspartate group to the proline. So they kind of add an additional, remember proline's got an additional sort of circular shape to it, and you can add on there using chemistry an extra aspartate group, or in this case, this insulin Lispro part. They've added a lysine and a proline into this sort of a mirror image of these two amino acids. And by modifying these, they can actually, come to think of it, that may be wrong. Maybe they just made the substitutions. I actually can't remember if they've actually added these on or made the substitutions. Chemically, it would be difficult to add a proline to a lysine without doing another peptide bond. So my guess is these are substitutions, but I've forgotten that, to be honest with you. This detamir insulin is shown here in gray, where again they've made this modification of this glycine to alanine. But you can see here by tweaking the amino acid sequence and slightly modifying, they can dramatically alter binding, stability, and folding and off rate of these of insulin. So it's a good example of structure-based drug design in action, which is very popular and hot these days. Now, one of the key aspects of duration of action of synthetic insulin has to do with the ability to, when you make it, the protein, you turn it into a powder or a crystal. And then it's important to be able to dissociate this crystal and dissolve it for drug delivery. So this was a major aspect for modification of these amino acids to make it so that the crystals could be quickly dissolved and dissociate into a usable, functional peptide and protein. So this is just an example of the modifications and substitutions that were made. And yeah, it is substitutions, apologize. So they were amino acid changes. And this is just an example of those and where those positions are and all the different drugs on the market with those. You can see, and for this one, this detamir is quite interesting, where they've just added a myristoyl group on this B30 in addition to what we talked about before. And these are the rapid-acting ones, so you can see B3, B28, B29 clearly modulate rapidity, and then the long-acting ones, in part due to probably stability and not getting degraded by adding that myristoyl group to protect it. What does that mean, long-acting versus short-acting? Well, you can see here detamir is over 24 hours in activity, and whereas this rapid one for chronic or very quick injection use to save lives is only a few hours, right? So we have a quite dramatic differences in effect. Now, when we talk about injections, where do we do the injections? Typically, when I was in graduate school, my PhD supervisor was a type 1 diabetic. He always did it in his thighs here or in his lower abdomen. But obviously, these are the major injection sites. And then some people obviously do it in the buttocks for aesthetic reasons. But the problem is the challenges are sometimes you can get lipohypertrophy if you're always injecting in the same site. So you're sometimes supposed to alternate who does what. And then I just wanted to ask, does anyone know anyone in the family? Now, obviously, we don't have our clickers with us, but you'd be surprised, and the answer is often many people have or know someone with type 1 diabetes. What are some of the challenges associated with insulin therapy? Well, it's difficult to mimic physiological insulin secretion patterns, and that's because they come after meals, they come at different times of the day, and just showing that here, you can see after breakfast, after lunch, after dinner, you get different spikes in insulin levels, and obviously this will change depending on the plasma. And modulating this and doing this and trying to replicate this with just a single injection is difficult, right? And that was why eventually what was tried to develop were some of these pumps, these automated pumps that can more precisely mimic and modulate these levels. So again, a lot of the things that influence insulin and the challenges associated with insulin therapy are that the things that affect blood glucose can be obviously diet, what you're eating, the injection site can make a difference, how much delivery you're getting, temperature, if someone lives in a very cold place or it's someone in a very hot place, exercise, how much exercise the person's doing, if they did exercise on the day, illness, stress, alcohol, if someone's an alcoholic or drinks quite a bit, can be an issue. Menstrual cycle can play a role. And obviously, importantly, hypoglycemia. So as I mentioned to you, there was a lot of interest in developing these subcutaneous insulin pumps, and this was one of the first insulin pumps shown here. And as you can see, obviously, it was not very practical to be walking around with a giant pump on your back, right? So it was miniaturized, and this is very similar to what my graduate advisor used to use back in the 90s, and these days they're much even smaller than that. But obviously the cost is typically higher for these pumps. These days, less so, but it is not trivial. And because of this permanent injection site, essentially, you basically always have something inside you. There is a slight increased risk of subcutaneous infections to these patients. And obviously, it is a minor inconvenience for lifestyle. Certainly not as much as the consequences of not having insulin, but something to be thought about. So there's a lot of interest in alternative routes of insulin delivery, and one of the major ones was developed by a company in the U.S. and is now part of AstraZeneca, and that is the nasal delivery of insulin, so through just a spritz. Obviously, there's still a lot of interest in oral buccal insulin and trying to deliver that, and a lot of combinations and flavors therein and continue to be in development. The inhaled insulin that I mentioned, either through nasal or through an inhaler. There's a subcutaneous route through different pens, and obviously the pumps. And you can get, obviously, a transdermal these days. There's transdermal micro patches that have tiny little needles as well, trying to inject them. So there are a lot of varieties to try and achieve insulin injection and delivery. So how do we monitor glucose? And one of the connections that is very important is constant monitoring, so we know what those are. And typically it involves a glucose monitor. You can also monitor when you go to the doctor. HbA1c is one of the things that they will measure, and it's an important readout. And then obviously the ketones that we talked about, using either a meter or just a urine test strip. What is this HbA1c? HbA1c refers to, it's a glycated hemoglobin, which identifies the average plasma glucose concentration of an individual over roughly the last 3 months. And this is why doctors like it, because it gives them kind of a snapshot of the last 3 months of glucose levels. It's often commonly referred to as A1C. It develops when hemoglobin become glycated or glycosylated, and so it's a good readout for how much sugar is around in the blood. Because the average lifespan for red blood cells is roughly 120 days, it does reflect a nice average of blood glucose levels over the duration. So A1C indicator, just to give you a rough sense of when it becomes an issue, this is a common sort of readout for when it's a problem. So for people with type 1 diabetes, it can feel basically a lifelong struggle to monitor both their diet, check their blood sugar, and administer injections. And then there's a lot of interest in terms of blood glucose monitoring and developing. So I know Huawei is developing a blood glucose monitor for the watch, for smartwatches and smartphones. Using also the camera. Apple is also developing. I think Samsung is also developing. So there's a lot of interest in trying to make it very easy for people to follow and check their blood sugar levels and use it kind of a smart app. Now, finally, in terms of treatments, one of the hopes is that we can deliver and provide new beta cells to patients, so using eyelid transplantation. So the idea is basically create islets or beta cells, grow them in the lab, isolate them, grow them in the lab, and have them to be able to be transplanted and survive enough to recapitulate a functional pancreas and Langerhans for patients. And the challenges are islets are desperately in short supply. It's basically difficult to get. The new sources of insulin-producing cells are required. Typically, transplanted cells run out of steam, so they don't last forever. And so you need to be able to you want the patient not only to have these, but also to be able to make their own, rather than having to constantly give a transplantation. And then obviously, just like with any cell therapy, you risk the rejection of those cells by the patient, which could lead to obviously dramatic issues of sepsis and rejection for the patient. So what are the ideal beta cells for transplantation? That would be capable expansion to useful numbers so that are actually producing a usable amount of insulin. That would be glucose responsive to insulin secretion. Insulin secretion would be glucose responsive, so they could actually perform at physiological levels. Able to control proliferative capacity. Obviously, you don't want them to overtake the pancreas and go crazy. You don't want them to be recognized by, in the case of type 1 diabetics, you don't want them to be recognized by those immune response. And then obviously this is where the stem cell therapy side of it becomes very, very attractive. So we want to be able to drive human embryonic stem cells towards new beta cells. And this can be done certainly in the dish, and then the question is trying to get them to then produce insulin and perform as desired. And challenges become purifying these, right? So even when you do it in a dish, you don't get 100% beta cell production. You also want them fully functional. You want to be able to control their growth, as I mentioned, and you want this transplantation to be safe so you don't get an immune response. So then you have to think about your donor and matching and donor matching. So future treatments are going to require improved islet transplants. And there's a lot of interest in artificial pancreas projects throughout the world, and producing new beta cells from embryonic stem cells. So not just taking them from donors, but actually producing them in the lab and then delivering these. And then finally, there's a lot of interest from not necessarily using embryonic stem cells, but can we de-differentiate adult stem cells back and make them useful for transplantation? So, we made it through. These are the things we've covered, and I think I've covered what I expect you to know, but generally you should be able to compare and contrast type 1 and type 2 diabetes, because those are important details to be able to know. All right. Thank you very much, guys. I'm really having trouble with my QQ. I don't understand. I can't log in. I can't reset the password because it's on Nancy's phone. If you want, just for today, email me any questions you have, and then I hope by Monday I've got it sorted out with Nancy on the QQ, because I like the QQ. It's nice to chat with you directly. Anyways, thank you very much.